High-flow nasal cannula, or HFNC therapy, is a non-invasive form of respiratory therapy in which heated and humidified flow is delivered to the patient via nasal cannula geometry. We have two example geometries, so these are the geometries studied in the work today, shown in the top right. Now, there are multiple proposed benefits of HFNC therapy, so improved oxygenation, moderate to standing pressure generation, and then also CO2 flush, which would mitigate or minimize CO2 rebreathing. And that is the effect that we're focusing on today, which is minimizing CO2 rebreathing. Quickly going over the boundary conditions for the model. First, we have the airway inlet. Now, inlet is in quotes because it functions as both an inlet and an outlet, depending on where we are in the breath cycle. This was a transient model, and we applied a breath cycle using user-defined function at the tracheal inlet/outlet there. Now, this controlled volume flow rate, so setting where you were in the breath cycle, but also species concentrations. So during the inhalation period, the UDF would track the species concentrations leaving the domain via the base of the trachea here, and it would accumulate the total volume of inhaled species during the inhalation cycle. Then at the end of inhalation, some of the oxygen would be converted to CO2 as a surrogate for cellular respiration, and then those final species concentrations would be applied to the next exhaled breath. The cannula inlet is a steady input of 100% oxygen at variable therapy flow rates, so we'll cover some different therapy flow rates today. And then the environment outlet is a pressure outlet with a species feedback of 21% oxygen, so it's essentially just ambient air. Now, there are going to be some contours throughout the presentation, so I wanted to show an example setup. They're all collected from profile views. Most include the patient facing towards the right side of the screen, as shown to the right. And contours were collected on multiple planes to provide a more complete view of the airway, as one single plane it may appear that certain pathways abruptly end when in fact the nasal passageway is just kind of going into or out of the page. So if we increase the transparency here and replace it with a contour, this is an example of some of the contours you might see. And this is mass fraction CO2, where red is high CO2 and blue is low CO2. So basically we're going to want to see as much blue as possible on the contours. Now, covering some previous work we did, we varied the mouth open percentage for a variety of models and tested some different cannula geometries, but all of the cannula geometries we tested in the previous work were dual prong geometries. And the main distinction between them was the inner diameter of the prongs. So both of these are the same cannula geometry, and what's varying is the mouth open percentage of the patient. So we can see at dual 100, so it's a dual prong cannula with 100% mouth open. We see a lot of blue in the upper airway. By end exhale, these are 3 separate timestamps throughout exhalation. So it's the moment at which the patient is exhaling at the highest volume flow rate, and then it's the moment that the most CO2 is entrained in the airway, and then end exhale. So end exhale is a strong indicator of how much CO2 the patient is likely to re-inhale. And we can see with a 100% mouth open, there's very little CO2 entrainment in the uppermost portions of the airway. And then by end exhale, even the oral cavity is mostly flush with that CO2. But if we close the mouth to 20% open, so this was kind of our intermediate difficulty flush scenario, we see significantly more CO2 entrainment, and then kind of this high CO2 front where we have expiratory flow breaching the uppermost portions of the airway, and then the therapy flow kind of combating that, and you create almost like this stagnation region back here and this front. And then by end exhale, there's a higher CO2 entrainment, so the simulated patient is likely to re-inhale more CO2. Now, what the proposed mechanism to combat this increased CO2 retention is a single prong cannula. So these are path lines colored by mass fraction CO2, and for the dual prong, you can see the therapy flow is being injected into the upper airway, but then it's not really reaching not much of it is reaching the furthest back portion of the airway and mixing with this expiratory flow, which that's what we want. So for the single prong cannula, we can see much more of the therapy flow is traversing the back of the nasal passageways, reaching the nasopharynx, and then it's going to mix with the expiratory flow. You can see the mass fraction CO2 increasing. I know they're a little bit small here, but then it's going out the open nair. So that's basically the proposed mechanism for how this is going to increase CO2 flush in difficult to flush scenarios. So now showing a plot of the mass of CO2 inhaled versus the therapy flow for both the single prong cannula and the dual prong cannula, we can see that the single prong cannula consistently causes lower CO2 inhalation for each of the therapy flows tested. And now these values were collected over multiple breath cycles. So each of these models was run for about 10 to 12 breath cycles, and then the first few were not included in the final breath averaged values because they were kind of traversing to their quasi-steady state, and then only those quasi-steady state values were included for the final metrics. Now, if we move from this intermediate difficulty flush scenario to the most difficult flush scenario in which the mouth is completely closed, we wanted to see how each of the therapy types responded. So CO2 inhalation values are tabulated here for the single prong and dual prong cannula at both 20% mouth open and 0%. And the dual prong cannula showed a greater drop in effectiveness when the mouth was closed. So for the single prong, the CO2 inhaled increased from 2.06 milligrams to 2.75 milligrams. So we had about 3/4 milligram increase in inhaled CO2. But for the dual prong, we had almost 5 milligram increase in CO2 inhalation when moving from 20% mouth open to 0%. So the dual prong cannula struggles significantly more in the closed mouth scenario. Now moving on to looking at some strain rate contours. We have a couple important regions circled. So if you remember back to the previous work where we had that stagnation region for the dual prong cannula with the 20% mouth opening, we can see that the strain rate in that region for the dual prong is quite low when compared to the single prong cannula. So we have increased mixing in this region as the single prong therapy flow is able to traverse back to the nasopharynx and then out the open nair there. Now, if we switch our focus to just outside, kind of around the cannula prongs, we can see that there's increased mixing for the dual prong, increased strain rate for the dual prong next to the cannula. So for the single prong, the cannula is in the left nares. We have increased mixing as more of that therapy flow is kind of turning around and being forced back out the nair because it's not able to cross that high CO2 front. Whereas for the open nares, for the single prong cannula, the right nares, you can see significantly higher strain rate, as this is where all of the mixed expiratory and therapy flow is evacuating out the airway. Now here we have some path lines colored by airway residence time that were released from the airway inlet. So what this is tracking is expiratory flow, and what we're looking for is to minimize the airway residence time. So we want to get that expiratory flow out of the airway, because that's our CO2 rich flow that's going to cause higher CO2 reinhalation if we can't evacuate that. So the single prong cannula showed significantly faster, or I should say, it showed faster, consistently faster is the word I was looking for there, evacuation from the upper airway, where the CO2 rich exhaled flow doesn't get caught in the upper airway quite as much. You can also see that all the path lines here are leading the open right nares, as there's no cannula prong here. So for the dual prong, we see higher residence time, so these path lines are getting up into that red region here before some of them are able to escape out either of the nares around the cannula prong. Whereas for the single prong, we see a pretty consistent flow path where the expiratory flow is either leading out the partially open mouth or just going straight out the open right nares here. Whereas there's none leading the left nares because that's where the therapy flow is being injected. In conclusion, we use CFD models to analyze the effect of asymmetrical high flow therapy on CO2 flush. And at intermediate mouth openings, the single prong cannula consistently resulted in less CO2 inhalation than the dual prong cannula. And then if we further close the mouth to be completely closed, the disparity between these two was heightened, so that the single prong cannula further outperformed the dual prong. The single prong cannula also resulted in lower residence times, so it indicated that the CO2 rich expiratory flow was more easily able to evacuate the airway. Thank you very much, and are there any questions? Yeah, that's a great question. So we started with, so I mentioned at the beginning that Vapotherm was the funder of this project. They have an experimental setup using an ASL 5000, so a breathing simulator, and they 3D printed an airway to exactly match our setup, and they collected oxygen information, and then we validated against that information. We also had a mesh independent study, time step independent study, and we tested some different solver settings. But in terms of actual experimental information, it was like a bulk oxygen metric.
There is PhD papers out there on nasal capacity if you want to. So I suggest you get Spence, Van Hove, or Adams. 2008 to 2016 there's a bunch of external papers that you used as well.
Okay, yeah, that's
DOE data, DOE data as well.
okay. That's really good to know. Thank you. Actually, if you'd like to talk after, so I can get some of those references, that would be great. Thank you.
Are there any other questions? I have one quick question. So you did not simulate the hit flux, right?
No, sir. No, there was no temperature considerations in this.
Do you think there could be an impact on the carbon dioxide concentration changes if you do include it?
That's a good question. That isn't really something that we didn't anticipate that would have a significant impact on the carbon dioxide concentrations. But that's definitely something that we could look at in